Hey guys, how's it going? So, I wanted to go over some topics that I think you may have reviewed before I came along because they are going to appear on your Unit 1 exam. So, I'm going to discuss characteristics of living things, levels of biological organization, and a little bit about history of science and the scientific method. So, okay, let us begin. All right, so welcome to biology. We did this assignment together. So, how did scientists discover cells? This assignment goes over how science is an iterative process. So, science changes over time as technology improves. As a result of science, then that technology can be used to further the scientific process. Science is an inquiry based process. We try to stay away from the scientific method, but it is a good way to describe different parts of the scientific inquiry process, but we use science to question and in an ordered way examine the natural world. So I will be posting a separate review on some important vocabulary words important to the history of science, the scientific method, and like validity and sampling, which I'm not sure if you guys covered, but that will be on the Unit 1 exam. So I will be posting a supplemental assignment on that. So we did this together. So, how have microscopes and the discovery of the cell advanced science and medicine for humanity? So, microscopes were a technological innovation as they improved and could see smaller things. Humans could see smaller things. People that had theorized that there were such things as cells finally had proof and they could see the cell. And how has this advanced science and medicine for humanity? These are just some examples. So, the germ theory of disease. Louis Pasteur and previous doctors had theorized that there were such things as germs, and the original doctor who theorized that there were germs actually wound up in a mental institution because saying that there were tiny things all around you making you sick, and especially insulting doctors because uh, birth became a medicalized process at that time. So, doctors that had previously not been involved in birth, which had been handled by women in the home, like midwives, all of a sudden doctors were involved, and doctors were going from cutting up cadavers, dead bodies that may be infected and also just literally dead and rotting, and going and delivering babies. And women were dying of infections at much higher rates than they had previously. And that was an insult to doctors to say that their hands could possibly be dirty and they needed to wash their hands. But without microscopes and the ability to confirm, like, oh, there's germs there, uh, Louis Pasteur would have also probably been locked up in the insane asylum. Speaking of Louis Pasteur, pasteurization, which is apparently a controversial thing today, should not be, was invented because we could see that there were microscopic organisms in our milk and other foods, and that they could be heated away and killed. As a result, too, vaccines were developed, such as Pasteur keeps coming up with rabies and anthrax, and further any other vaccine, especially mRNA vaccines like the COVID vaccine, wouldn't be possible because we wouldn't know anything about the cell and its genetics, DNA, without microscopes. So, speaking of DNA, right, even smaller than the cell itself is its DNA. And James Watson, Francis Crick, and a very important and sometimes forgotten Rosalind Franklin discovered the structure of DNA in 1953. This would not be possible without the discovery of the cell before that in 1665. And later, genome sequencing, which started in 1977 with the bacteriophage and finished with the mapping of the human genome in 2001, would not have been possible without that discovery of the cell in 1665. So, something that happened in 2001 was affected by something happening in 1665. This is what I mean about how science is a process that builds on itself, and sometimes we find out that our previous answer was incorrect. We didn't have enough information, especially with the use of new technology. We can either confirm or refute theories from the past. And recombinant DNA technology, DNA splicing, none of this would be possible without Robert Hooke in 1665 discovering that cell. So, microorganisms are organisms that cannot be seen with the naked eye and can only be seen from the microscope. And we would not know that microorganisms existed without the technology of the microscope. And there is technology today that will lead to widespread knowledge, such as our widespread knowledge today of germs and cells, that is not considered a revolutionary topic for the general public to be knowledgeable about, but would have been very revolutionary in the 1300s, absolutely unknown, and even in the 17 and 1800s, not as commonly known, especially germ theory did not come around until almost the 1900s. So we are living in an age of widespread scientific knowledge and literacy that has not been known in the past. And this is partially due also to the ability to spread scientific knowledge through technology like I am doing right now. So good job, humans. So the microscope, thank you, microscope, for making microorganisms visible to us all. All right, so now let's talk about the characteristics of living things. What do the subjects of all of these images have in common? We've got cactus, macaw, parrot, kittens, a little husky, Billie Eilish, and some bacteria in a petri dish. And all of these things are alive. So it's alive. From the tiniest bacteria to the largest blue whale, all organisms on Earth are alive, right? If it's an organism, that means it's a living thing. But what does it mean to be alive exactly? How is this a category that could encompass so many different things? Have you ever wondered why you get hungry or what is happening when a scab forms on your knee? These processes and many more happen because you are a living thing. So, what is life? The definition of life is charged with meaning and often controversial. Some of the most passionate debates, for example, over stem cells or the right to die, revolve around the definition of life. Whether talking about when life begins or when it ends, most people have a clear idea of what they mean when they talk about life. However, it often turns out that people mean different things by living. Life is often defined by what we know, thus, it is often easier to say what life isn't. Furthermore, there is an exception to almost every general feature that might be thought of as characterizing life. And that's important because you might see six characteristics of living things, seven, eight, and they're all talking about the same characteristics, just at different levels of nuance. And so we're talking about this at an appropriate ninth, 10th biology high school level. But if you were to take an AP biology class or in college take biology classes, they would discuss these same characteristics just at a higher level of complexity. All right. So, which of the following terms describes all living things? Is it A, atoms, B, molecules, C, organisms, or D, celestial bodies? I feel like Dora the Explorer. What's the answer? It's C, organisms. All living things are organisms. So, what is an example of an organism? You can see some in the picture here. We have, I think, some fish, an eel, maybe, or a sea worm, a butterfly, a rat, a squid, some seaweed. We are organisms, dogs and cats are organisms. So, what do all these organisms have in common? Because bacteria is a living thing. So, what do people are considered widely the most complex, perhaps, maybe because we're conceited. But what do we have in common with bacteria? Hmm. Well, let's take a look at this baby alive doll. It looks pretty similar to a real baby. It grows, it eats, it even poops. But of course, we all know it's not a real baby. But why? What characteristics set a real baby apart from the doll? So, this doll is made out of plastic, for one, right? We know living things are made out of cells, they're made of organelles. So this baby is not made out of cells. We could leave this baby in a toy box and nothing would happen to it. We could leave it there for years, in fact, and it wouldn't wither away. You could walk into a wall with this baby and it wouldn't cry. And even though it can grow and poop and you can feed it, it's not changing based upon any of that input or output, right? Like a living thing would be responding to those stimuli. So we know this innately, what things are living and what things are not in general. But how do scientists characterize living things? So we're going to fill this in as we go through the characteristics, starting with, oh, look at this beautiful example of living things. So we're talking about bacteria, amoebas, fungi, snails, raccoons, seals, more mushrooms, a Zapothos plant, Crowd of people, flamingos, we're all alive. What do we have in common? Number one, we're organized. So all living things have one or more cells, and unicellular organisms have one cell. And they have specialized structures that function to help the unicellular organism survive, such as an amoeba or a prokaryotic cell, which would typically always be unicellular organism, and that would be like a bacteria. And multicellular organisms, like humans, which have more than one cell, have many types of specialized cells that are organized into groups based on their functions. Like this picture here. So, a multicellular organism like a human, we have many different specialized cell types. But a prokaryotic cell just has different organelles that function to keep the organism, the prokaryotic cell, alive. And there are unicellular eukaryotes, like protists, and they also have specialized organelles, not specialized cells, right? Because they're just one cell. So a unicellular or single-celled organism is made up of only one cell. So examples can include yeast, protists, amoebas, and all bacteria. Many unicellular organisms are prokaryotic. So what are some examples of unicellular organisms? Well, have you ever had a bacterial infection in your throat, like strep, streptococcus? That's a bacteria. MRSA, that's a bacteria as well that can give you a terrible bacterial infection. And those are just some examples of prokaryotic organisms you might interact with in your life. Hopefully not MRSA, but probably for mostly everyone, strep. And this is what antibiotics do. They go against living organisms, bacteria, and that's contrasted with antivirals. And viruses, excuse me, being alive or not, is a hot topic of debate in science. But typically, we consider viruses are not alive, and antibiotics then go after the living organisms, bacteria, that might be in your life. And you might also take probiotics to increase the bacterial populations in especially your gut, and that's pro-bacterial development because there are good and bad bacteria. All right. So, what about now? Moving away from bacteria, unicellular organisms, and talking about the multicellular organism, us that's colonized by bacteria all over. Multicellular organisms are made up of many cells. Most organisms that we can see with our naked eye are multicellular, and we call these typically eukaryotes, which include plants, animals, and even fungi like mushrooms, are all multicellular eukaryotes. So, what are some examples of multicellular organisms? That tree, you and me, dogs and cats, a snail, a tiger, lions, tigers, and bears, in fact, are all multicellular. So uni meaning one, multi meaning many. So this is a chart of prokaryotes versus eukaryotes. So you can see in the domain of eukarya, we have protists, which are single-celled eukarya, and plants, animals, and fungi, which are typically multicellular. And then we have here in the domain of prokaryotes, bacteria and archaea, which are single-celled prokaryotic cells, which are like the protocells, the early cells, and they have no nucleus. So cells are the basic unit of life, and they come in two varieties. Much more frequently do we interact with eukaryotic cells, which include plant and animal cells, but also there are prokaryotic cells. So prokaryote means no nucleus. And these unicellular organisms have no membrane-bound organelles. So that means no mitochondria, no endoplasmic reticulum, no Golgi body, no nucleus, no membrane-bound organelles at all. Prokaryotic cells are enclosed by a cell membrane, and many also have a cell wall for structural purposes because they're single-celled. These cells carry genetic information in DNA, yes, common to all living organisms, but it is not contained within a nucleus, no nucleus. So the DNA is floating freely in the cytoplasm, usually in a ring. Most prokaryotic cells have ribosomes because making proteins is what living things do, baby. And all bacteria are prokaryotic cells. So you do interact with prokaryotic cells all the time. It's just you can't see them because they're so small. Eukaryotic cells make you. So that's how you can remember prokaryotic, protocell, early cell, eukaryote, you. You are like an animal cell. You are an animal. You are a eukaryote. So are plants and fungi. Eukaryotic cells have DNA enclosed within a nucleus. So that's what differentiates prokaryotes from eukaryotes. Prokaryotes have no nucleus. Eukaryotes have DNA within that nucleus, which also has membrane. And eukaryotic cells also have all of those specialized membrane bound organelles, which perform life functions for the cell, which we talked about, such as the nucleus and the mitochondria, the cytoplasm, which is present in both cell types, cell membrane, which is present in both cell types, but all those membrane bound organelles not present in the prokaryote. Here's how they look under the microscope. You can see these are plant cells characterized by their connected cell wall and organized structure. And look how much larger they look under the microscope than those tiny little prokaryotes, those bacteria. They're teeny tiny, and each one is an individual living organism, whereas these cells are organized into tissue of a larger plant, I believe in that case, an onion. So, biological organization, uncharacteristic of living things, what we're talking about. Living things are highly organized, meaning they contain specialized coordinated parts. All organisms are made up of one or more cells, which are considered the fundamental units of life. So, if we're looking at levels of organization in biology, we can start before life. We can look at the atom. So, molecules are made up of atoms. So, water, for example, is made up of two atoms of hydrogen and one atom of oxygen, H2O. A macromolecule is a larger molecule like DNA or glucose, which is not alive, but it is a larger molecule. We're going to talk about macromolecules and biomolecules after we finish with cells. But then now, after this point, after the macromolecule, when we get to the cell, and another macromolecule, for example, that you would be familiar with is protein. So then we get to the level that is alive. So between macromolecules and cells, we could put organelles, right, which are not alive. They are functional units of the cell, but they themselves could not sustain homeostasis. They don't meet all the characteristics of living things like a cell does. A cell is the smallest functional unit of life. And after that point, then we have a cell, which is a collection of macromolecules and organelles that are organized in a fashion in order to carry out homeostasis, use energy, reproduce, and meet the characteristics of living things. Tissues, which are a collection of cells, organs, which are a collection of tissues that carry out a specific function for the organism, an organ system, like your respiratory system, which consists of organs like your lungs that carry out a specific function for the whole organism, like you or your cat. And then we can go even higher to a group of organisms is a population, a group of organisms of the same species is a population, a group of multiple different species living in one area is a community. The interaction between those different species and their non living environment is an ecosystem. And then all of the life on Earth is the biosphere, right? So, this is why biological organization is important because we can look at life in this way from less complex to more complex, the whole planet Earth. And human beings, we just really like to organize things and make hierarchies. And this is one that is important for you to know in biology. All right, so cell theory. The cell says, and we are all made up of tiny units that I call humans. The other cell says, interesting theory. But we know that that's not right. So, cell theory says that all organisms are composed of one or more cells. All cells arise from pre existing cells, and this cell is the structural and functional unit of all living things. And we can see the cells that make up tissues, that make up organs, that make up the organ systems, that make up the organism. The cell is the structural functional unit. All of these components are made up of cells themselves, but the cells organize into tissues and organs, organ systems, and organisms. And interesting fact, each cell has the exact same DNA. And the cell, when it makes a copy, knows which parts of the DNA to copy to make a liver cell, a stomach cell, a cheek cell, a hair cell, a lip cell, a brain cell when it needs to copy. So, this is something that is fascinating about our DNA and how it is used to build proteins, baby, in different parts of the body. All right. So, how do we know that all cells arise from pre existing cells? So, looking back all the way to 1668, a contemporary of Robert Hooke, Francesco Redi in Italy, had wide mouth jars containing flies and meat. So, why did he do this? So, There was a theory at this time that spontaneous generation of maggots happened from meat. So if you leave meat out, spontaneously maggots appear, meat turns into maggots. And Francesco Redi said, no, 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 no. What's happening is flies are landing, they're laying eggs. And when those eggs mature into maggots, we're seeing the maggots in the meat. So he left this first jar as a simulation of that typical condition of people leaving meat out. It's an open mouth jar, flies coming in, laying eggs, maggots appear. So that shows the maggots are coming from the flies. So now he created a simulation where no flies could get in, and he covered the jar in gauze. So in this simulation, through the gauze, small materials, it's a semi-permeable membrane, small materials like the eggs could still drop through the gauze. So even though no flies entered, they still managed to lay eggs in the meat, which hatch maggots. And boom, maggots in both of these conditions. However, Francesco Redi said, ha, so we see here maggots, maggots, meat, maggots connected, but hear me out. Meat is not made of maggots. Let's seal the jar. He sealed the jar. No flies could enter, so no maggots hatched. No eggs could enter because no flies could enter. The sealed jar did not turn into maggots. So spontaneous generation of maggots was not happening from meat. All cells arise from pre-existing cells. Where did the first cell come from? That's a more complicated question. But spontaneous generation is not true. Fun fact, my grandfather, born in 1932, believed that meat turned into maggots and told my mom this when she was a kid. So this was a belief that persisted long after Francesco Redi's experiment in the 1600s. Coming down now to the 1800s and bringing up Louis Pasteur again. He keeps coming up, very important scientist. He took this a little bit further and not just looking at organisms we can see with the naked eye, maggots, let's look at bacteria, organisms we can't see with the naked eye. So he boiled broth in various flasks for 1 hour to sterilize it and then allowed it to cool, drawing in fresh air. In the first condition, the open flask allowed air and any bacteria present in the air to enter. So in this condition, like Francesco Redi's open mouth jar, it was contaminated with bacteria, the broth goes bad. In the second condition, a cotton plug filtered bacteria from the air entering the flask. So, makes sense. The cotton plug is more effective at filtering microscopic organisms than the gauze was at filtering the flies, dropping their eggs. Also, may have to do with the width of the flask versus the jar. But then Louis Pasteur said in this uniquely shaped flask, which draws in more air, removing the bacteria from the flask, allowing them to settle in the long neck, he was left with a sterile broth. So there is not spontaneous generation of bacteria or even mold, for example. If you have moldy bread, there was a mold spore in that bread package. So the mold doesn't come from nowhere. Bread doesn't turn into mold. There was a mold spore in the package. And so we know this because we have done many experiments to disprove spontaneous generation. This is why cell theory is a proven theory in science. So in science, that word theory has a little bit of a different definition than it would in common parlance, common use in English. Like, I have a theory. When we talk about theories in science, it means that this has been uh, proven to be true and accurate in many experiments. There are no experiments that disprove the theory. If you were to have an experiment that went against an established scientific theory, it would be breaking news. It would be all over the place. Like, if you can disprove spontaneous generation, like, report it to me. We're going to get on the news together. But scientific theories like evolution, for example, or cell theory that we were just talking about, are established theories. The Big Bang Theory, this means that there is a preponderance of evidence. Every experiment that is questioning this theory has proven it to be true. There's never been an experiment that says it's not true. If there was, there would be, it wouldn't be a theory. All right. So, talking about unicellular and multicellular organisms, even unicellular organisms are complex. Inside each cell, atoms make up molecules, which make up cell organelles and structures. In multicellular organisms, similar cells form tissues. Tissues, in turn, collaborate to create organs, which are body structures with a distinct function, like your heart. Organs work together to form organ systems, like your circulatory system or your digestive system. Multicellular organisms, such as humans, are made up of many cells. The cells in multicellular organisms may be specialized to do different jobs and are organized into tissues, such as connective tissue, epithelial tissue, which is like in the inside of your mouth, muscle tissue, and nervous tissue. Tissues make up organs, such as the heart or lungs, which are organized into organ systems, which carry out specific functions needed by the organism as a whole. All right, these are some examples of multicellular tissues in humans. And a unicellular bacterium is not going to have tissues because it's just one cell. So we're going to run a little long if I go through all the characteristics of living things in this video. So I think we'll finish with this review of levels of biological organization and pick up with the rest of the characteristics of living things in a second video. So we only got through one, but an important one, organization. So, levels of cellular organization, again, going all the way back, the atom, the molecule, the macromolecule, the organelle, and our cells are made up of organelles, which are functional parts of the cell that carry out a specific function. Our tissues are made up of cells, which are functional units of the tissue that carry out a specific function to help the tissue carry out its function as part of the organ. And organs are made up of tissues that work together to carry out a specific function for the organ system that makes up the organism, which has all of these different organ systems, organs, tissues, cells, and even organelles working together to maintain homeostasis and carry out all of these life functions that are involved in being alive that we're about to go through. So, plants are also organized in this way. Other animals are organized in this way. Cells, tissues, organs, organ systems, and the whole organism. We can look at plants this way, but we're not going to talk about that too specifically because we'll use humans as our example. And this is going from least complex, which you can think of as smallest, to most complex, which is largest. So that's the whole organism. All right. So this I think I'm going to give you guys as part of your exam review. But yeah, I don't have the answers plugged in. So I'm going to give this to you guys as part of your exam review. But this is just a vocabulary review of all the different parts of the levels of cellular organization, organelle to organism. So, which diagram best represents the levels of organization in the human body? All right, Dora the Explorer again. You have a reference here on the left. Is it one, two, three, or four? So the smallest one should be in the middle and the largest one should be on the outside. All right, the answer is two. So cells are the smallest, followed by tissues, which are made up of cells. And this diagram helps illustrate it. Organs, which are made up of tissues, which are made up of cells. Organ systems, which are made up of organs, which are made up of tissues, which are made up of cells. And then if we labeled this outer circle here, what would we write in it? Organism, because the organism is made up of organ systems. All right, so we'll stop here and we'll pick up in part two with growth and development. Hope you guys liked that, and I will make some more videos also going through those assignments that you did before I came around because I do see that they were challenging. So again, we'll pick up with part two of characteristics of living things, growth and development in the next video.